I'm sorry.
That's all right.
So
I can do for you.
So I'm kind of confused about like what I mean by it will be trisomer for chromosome 21. So like
So what does tri mean?
like it has 3 chromosomes.
Mm-hmm.
Yeah. So like, but is it like those single strands or is it the chromosomes?
The whole chromosome. The entire piece of DNA that makes up a chromosome.
Okay.
So it looks like a chromosome, like that X thing during the middle of replication, but
it's not dividing.
That's right. That long, skinny little strand.
So, yeah, for chromosome 21.
Just 21. So
Yeah,
there'll be 2 of everything and then 3 of 21.
Okay. Yeah, because I was confused. Like 2, everything out, everything other chromosomes. 3 of 21. But then it says like an individual with a translocated piece of chromosome 21 plus 2.
Right. So normally, the only way that you get Down syndrome, or most commonly, you have 3 chromosome 21s. But it turns out that all you actually need is a small part of chromosome 21 to be present in 3 copies in order to have those symptoms. Okay? So most of the time, you'll see 3 actual chromosomes, but sometimes a piece of chromosome 21 will accidentally get stuck on the wrong chromosome. And so you don't have 3 complete chromosome 21s, but you have 2 complete plus like the nub that you need to make you Down syndrome.
And so then translocation is like
that's the name of sticking that extra chunk on a chromosome.
But non-disjunction is like translocation is like a part of that?
Translocation is the process, the name of the process that translocates. So again, think about the words, right? Trans, different, locate. So translocation is the process that moves that extra bit of 21 onto wherever it is, okay? And so you generate a translocation by translocating that section of 21 onto that other person.
Okay. Yeah.
Okay?
Yeah. Like, because I'm confused with like what the difference between like non-disjunction and translocation.
Right. So non-disjunction can be a thing that causes the extra chromosome. Okay. So non-disjunction is when the chromosomes don't separate.
Yeah.
So if they don't separate, that's one of the ways you can end up with an extra chromosome. That's not going to generate the translocation. Translocation is a different type of error in making the chromosome.
So if you have, you can have Down syndrome if you either have like 3 like chromosomes, or you can have like a piece of chromosome.
And if you have 3 chromosomes, then you probably had an error
during meiosis.
Right, with non-disjunction. And if you have 2 plus, then you have a translocation.
Okay.
Okay?
Yeah. So non-disjunction or disjunction does not equal translocation.
Right.
So you can have Down syndrome with if one of these 2 guys. So of 21.
I gotta start class.
Oh.
So you can maybe see if you catch me during the break, or you can go to Dr. Tajirian's office hours. She might be better suited to help you since she's the one who taught us. Okay. Thank you. All right, guys, I'd like to get started. So, couple things just to get the ball rolling. So, in case you don't remember, I'm Dr. Holtzman. I will be teaching most of the lectures moving forward. I know that some of you have got some questions about the first midterm. If you have questions about the first midterm, I recommend that you go to Dr. Tajirian's office hours. But if you have questions about the class as a whole, I'm happy to work with you on that during my office hours. I'm glad to see so many of you here today. I wanted to let you know that the campus is working on contingency plans because, as I'm sure you're aware, many of the private universities in the city are starting to close. And I want to explain why they're closing and why we have not yet closed, and what we will do for this class if the campus decides that we are closing, okay? So the first thing to know is the main reason that private universities are closing is, one, they are private. One of the things that is worth understanding is there is a wealth gap in our country, and one of the consequences of that is that private institutions have the money and resources and lack of consequences for closing. Public institutions are paid for by your tax dollars, and we need to do our best to provide you with the best standard of education we can provide. If New York City decides to close elementary schools, then that means that every elementary school child must be at home with a parent or with a grandparent, okay? So the economic impact is much greater than, for example, a private high school or a private university. So countries, nations, cities, everybody is trying to balance economic cost with health-related issues. Another reason why the private schools are closing is that their spring break is next week. And so they are going to transfer online this week so that the faculty can spend next week, while everyone's on break, putting all of the course material online and giving them time to make decisions about what's going to happen after spring break. Are they going to stay closed, or are they going to open back up? Our spring break is not for a month, okay? So we are not in a position right now where that has its global advantage, right? In addition, medically, and I'm going to walk through how this virus works, it is not yet advantageous from a health perspective for Queens College to close, okay? So that's the first set of issues. Second thing that's worth your knowing is that right now, because the campus is not officially moving to an online environment, the expectations are that you come to class and you come to lab. Now, I recognize that there are circumstances where that may be more challenging. If you are not feeling well, we don't want you here, right? Like, please stay home, okay? If you are the primary care provider or live in a home with someone who is at significant risk of this virus, that is another justification for potentially staying home. So starting today, I am going to be recording the actual lectures in a video format, and they will be available to you on Blackboard. They are hopefully synchronous, which means that if you are sitting at home right now, you can join me online, okay? To see how, and I have never done this, so you're gonna have to bear with me. That's why we're doing a dry run. In case we close next week, we'll already know how to do it, right? And frankly, you don't want to watch 3 hours of me at home online. You want to be here. But the way that it will look for you guys should be like this. So what you're going to see is this little extra thing here at the bottom on the side, and that will give you a link to today's lecture, which I'm not yet recording live. Once I hit the record button, you'll be able to actually jump right into that lecture. You can even type in and ask me questions, which I probably won't know how to answer while I'm lecturing, but we'll try. It will also be available for you to download. So if you decide, for whatever reason, that you would prefer not to be in the physical class today, you can join us remotely. And like I said, assuming it all works today, it'll be available for you to download, and you can also join us live during the recording moving forward. All right, I need you guys to be quiet. I know this is really exciting stuff. Now, labs. The lab policy currently is, if you miss more than 2 labs that you do not make up, you get an automatic F. I have been instructed by the university that I am allowed to be a little tiny bit generous, but I can't go too far in that because I also am responsible for ensuring that you get the same quality quality of education that you paid for. And we have designed the labs to provide you with experiences that are difficult to move online. So as long as the university is open, I need you to come to lab unless you are personally ill and at risk of sharing your illness with someone else. It's also very clear that class sizes of 20 or less are much lower risk than a class of this size, okay? So we'll talk about that in just a moment as well. So please do your best to come to lab. If you can't come to lab because you're ill, look at the standard policy around how to do a makeup lab. You can make it up in one of the other sessions that we're running at the moment. Again, you can miss 2. More than that is going to be a challenge. If we get moved online, we will, of course, do our best to give you the best possible experience of a lab at home. And we have some really good strategies for that. It won't be exactly the same, but it will help ensure that you have the skill sets you need moving forward. All right, so I also have heard from several students that their professors are not all as ready to go online as we are. If any of your professors seem at a loss, you can let them know that the Center for Teaching and Learning, which is in this building at Queens College, has people whose full-time job it is to help them move things online. And you can also send them to me. I'm the Associate Director of the Center for Teaching and Learning. I help craft the website and resources available to faculty to do that, which is also partly why we're going to try this out today. Okay, so let's take a quick chat and just talk about the virus, just so that this is a biology class. We can do that. Pardon? This is not recorded. We can, but I haven't put this in since I haven't figured out what I'm doing yet. We're going to play it by ear. I'm going to switch the lights. Oh, and the last thing just to point out is I normally dress up for lab class, but as a part of the contingency plan, today I had to learn how to feed all the animals in the animal facility because I have animals in the animal facility, and in case we're not allowed on campus, I now have special permission to come here, and since I was, feeding animals and getting wet, I dressed down for today. The life of a scientist, right? Okay, so the other thing just to keep in mind, as a part of this recording, I can't use a laser pointer because that is not recorded. I have to use the mouse. So we're going to do our best to try to see that mouse. And it also means I'm kind of stuck by the computer. So again, we're all in this together. So, all right, so the first thing that I wanted just to point out is that the virus is an RNA virus. It's a single-stranded RNA virus. Conveniently, in the second half of lecture today, we're going to be talking about these viruses. I can post the PowerPoints on Blackboard as well. I just haven't recorded it. This was initially identified as being in China. It was probably originally in the Chinese population sometime in the middle of October, maybe early November. But the first identified cases were December 1st, and the first recognition of an outbreak was December 31st. It's a common type of virus. There are many of them. They cause the common cold. You've all had versions of this all the time. But there are a few examples of variations of this virus that are relatively dangerous. They're dangerous because they kill lots of people. This one is not very dangerous. Okay, I'm just going to put that out there. But we didn't know in December when it was first identified how dangerous it was going to be. So to give you an idea, right now, as of today, there are 113,000 known cases, of which there are about 4,000 deaths. Now that sounds like a lot, but right now we're only testing sick people. So that means you have to be sick enough to go to the hospital to get tested. And so we think that this is going to be difficult in terms of mortality rates for less than 0.5% of the population, probably closer to 0.1. And that's in contrast to SARS, which some of you may have heard of before. It's another one of these viruses. That has a mortality rate of between 10 and 20%. And again, different than Ebola, which has a mortality rate of 50%. Okay? So the reason this virus is of concern is not so much because it is so dangerous for an individual who gets it, it's that none of us have had it before. So you now have the entire world who's going to catch the same cold in a short period of time. It is predicted that in the next 3 to 6 months, 70% of the global population is going to get this. Okay? Now, what is this? Right? Now, that's in contrast to like the flu, right? The flu, like 20% of the population gets in any given year, but every year there's another variation that comes around. So this is what the viral particles look like. So the virus has the protein, these proteins on the outside, and then it has its RNA in the middle. It is highly likely that this virus came from animals in the Chinese wild, possibly through food consumption or marketing, but it's very likely an animal origin. There's a whole host of people talking about how this is some laboratory made this thing and put it out in the world. It's a bioweapon, and some people are saying China did it, and some people are saying Iran did it, and some people are saying the US did it. No. Just no, okay? First of all, it's not hard to make. Okay, it's really not. I could do it in my lab, probably. I'm not going to try. I don't have time for that. But there's nothing that's a biological advantage to something like this. Okay, if you were going to make a bioweapon, it would not be this. So I'm not saying there aren't people doing weird things in labs they shouldn't be doing. This is not one of them. There's no evidence for that. There's no logical reason for that. Okay. So what are the symptoms of this virus? How do you know if you have it? What should you be worried about? So you may not be able to read all of this, but basically it's just a comparison of the coronavirus, this variation 19, with the seasonal flu and the common cold. And the most important thing here is that this is unique in that it makes you go from feeling fine to feeling really sick really fast. So the common cold, you're kind of like, oh, I feel a little sick. Maybe I didn't sleep enough last night. And then like 3 days later, you're like, oh God, I've got a runny nose. I feel terrible, right? This is like, I feel fine. Oh my gosh, I can't breathe, okay? So there's a lot of issues with lungs. In this disease, there's also not been associated with a runny nose. So if you have a runny nose, this is probably not what you have. If you think you have this, call your doctor's office. Don't show up, okay? They will tell you what to do. But if you show up, they're going to freak out because they're going to be like, oh my God, you might have just infected the entire office. So call, tell them you're coming. They will tell you what they need you to do to keep yourself and everyone around you safe, all right? Other things to know. We don't know exactly how long you're contagious. A lot of people seem to have no symptoms. When they have no symptoms, are they contagious? We don't know. Possibly, right? We think that you can be contagious for about 14 days, and we think that you tend to become sick within those 14 days. That's where that 2-week quarantine is coming from. If you aren't sick within 2 weeks, you're probably not getting sick, right? What else do we need to know? Most people get sick in about 5 days. So once you've been exposed, most people after 5 days, they get sick. And currently we think about 5% of cases, people get really sick. And what they get sick with is pneumonia, all right? And that's where the lung issues come in, and that's where the hospitalizations come in. So what are other things we want to be thinking about? So ideally, it would have been great to contain this virus, by which we mean that it's going to stay in China. That did not happen. It is now in a lot of places. And so the next best thing is to try to spread out the contagion, okay? So let's just do some quick back of the hand calculations. The World Health Organization thinks that 60% or so of the world is going to get this in this next year. Let's pretend it's 50% because that's an easier number to do math for, right? New York City has about 10 million people in it. So let's imagine that 5 million people are going to get this in the next 3 weeks because we do nothing for containment, okay? Now that means that even if only 1% of people are sick enough to need some medical attention, we need 50,000 hospital beds in the next 3 weeks in order to contain and provide the medical support for these people. We do not have 50,000 empty hospital beds, nor should we have 50,000 empty hospital beds. We don't want 50,000 empty hospital beds because that's a lot of money to be spending on hospital space that no one is using most of the time. So right now, the best thing that New York City can do is spread those cases out. So instead of everyone trying to go to the hospital in the next 2 or 3 weeks, everyone's going to go to the hospital over the course of the next 6 months to a year. And that's what's shown in this little diagram here. What we're trying to do right now is change this lots of people sick at the same time to less people sick at any given time and spread it out and give everybody, the doctors, a chance to provide everyone with the medical attention that they need. Right? So, what are the kinds of things that you can do to help? You can wash your hands all the time, right? You can stay home if you are sick. You can, if possible, stay home in general, right? Because you'll be less likely to be sick. You can be careful when you are with people who are at risk. So that includes people who are older. Okay, this is quite dangerous for people over 80, but it also includes people you may not recognize as being at risk. If you are immune compromised, let's say you had cancer and your spleen needed to be removed. Let's say you have rheumatoid arthritis, but you're 20 and you have to take immune suppressing drugs in order to keep yourself healthy. Let's say you've recently had a really bad asthma attack. Those are things that make you at risk, but someone else around you may not know that. So, other things. Wearing those masks doesn't help, okay? If you are sick and you sneeze, wearing a mask helps. It covers up the sneeze, keeps that sneeze from getting anywhere. If you are not sick and you wear that mask, it gives you a false sense of confidence. Studies have shown it increases the rate of infection because people are less careful. You don't wash your hands as much because you're like, oh, I'm okay, I'm wearing a mask, right? You touch your face more because you're like, oh, it's not sitting in the right place. And most people wear the wrong masks. So there are two kinds of masks that are generally available. There's the standard ones that surgeons wear. Those do not protect against the virus, okay? The kind you need are called N95 masks. They sell them at Home Depot for when you're painting. I don't know why, but I have a stack of them at home as a consequence. And those protect you. So if you are sick, you should wear one when you leave your house. If you are not sick, unless they change the advice right now, it's not a good idea. Aside from the fact that it scares the crap out of everyone else. Okay, question. Someone had a question? Yeah.
I heard that if a Japanese doctor said that if you hold your breath for like 10 seconds and you burst out into coughing, that means that you have it.
Right, so the question is, how do you know for sure if you have the virus? Right now, there's no way for you at home as an individual to know. Even if you have pneumonia, which is the thing that you're worried about in the context of this virus, you don't know whether the pneumonia is caused by this virus or by something else. I'm sure each of you has known somebody that's had pneumonia. I mean, my kids certainly have known their friends have pneumonia. So you may know if you are feeling sick. You certainly know if you have a fever, but there's no way for you to know if you have it. We also are not doing enough testing of healthy people right now or of everyone who's necessarily sick. So we don't have a good indicator yet. Maybe in a year or two, we'll have a better sense of what all the information is, but no, that's not accurate right now. Any other quick questions? Okay. So the other thing that's just worth knowing is who's at risk. So right now, there are almost no children there's 0 children under 10 who've died. There's only 1 person under 20 that has died. And the people getting sick is an even distribution, but the number of people dying, for whatever reason, it's very much impacting older people, particularly older men, older men who smoke. Okay? So those are the at-risk populations. So you as individuals are not likely at risk, though you can get sick. Okay, so that's my little coronavirus spiel and my little, we are prepared. And so it's, as an individual, you don't need to freak out. As, society, yes, we should do stuff. Trust the scientists. We're working on it. Okay, so now goes our experiment. This is what I see. Let me hit a few buttons here. All right, I am now recording this session. I believe I have a pointer. There are 2 other people logged in. Are the 2 people logged in to my video in the room? If you are, could you raise your hand? Okay, can you see my cursor when I move on your screen? Can you see the little cursor moving around? Yeah. Excellent, that means it's gonna record my little cursor. Now, the only thing that I think is a little frustrating with this system is that it's not as big on the screen that you guys are looking at. So if some things are too small, let me know, but that's one of the limitations of this, as well as those little join the system things. Those don't show up in the recording though. Those are just there while we talk. So, okay. So, whoa. Okay, we're gonna start today's lecture, and now you know how this will work if we are asked to run all our classes online. Okay, so today we're gonna follow up on what Dr. Tajirian has already taught you guys, but we're gonna be looking at the next steps. How do we go from DNA to protein? This is also referred to as gene expression. How do we express a gene? How do we make it do something? Whoa, now sound is on and it's beeping and it's all good. Okay. So can I move through the slides? How do I move? Oh boy, this is gonna be a fun adventure. It's not letting. The arrow? No, oh, it is the arrow. Look at that, you guys are awesome. Okay, so when we are scientists and we are studying how these different things work, we are going to use different organisms. And when we look at any organism, right, different organisms are gonna have different phenotypes. So if we look at you guys in the room right now, you're all humans, but you don't look the same. And you don't look the same because there are phenotypic differences between you, and those phenotypic differences are because you have different proteins, and you have different proteins because you have different DNA, right? And so we're gonna look at how you go from that DNA to those proteins, and I just want you to keep in mind that these are all things that researchers have discovered using different organisms to try to answer questions. And so we're gonna look at a few of those organisms today. Wow, that is much too small. All right, let's see if I can, oh, look at that. It's backwards, but that's kind of interesting. Okay, so the first thing that research scientists came up with in this process is this idea that you have a gene, and that one gene makes one protein. It's called the one gene, one protein hypothesis. So at this time, we knew that DNA was the genetic material. Yes, there was a time, okay, before I was born, but there was a time when we did not know that DNA was the genetic material. So, and we knew a lot of biochemistry. We knew that cells were able to carry out processes like the citric acid cycle that you learned about in mitochondria and photosynthesis that you learned about in plants. And so we could do biochemistry and say compound A becomes compound B. We also knew that there were proteins that carried out reactions, right? Those are called enzymes. And we knew that these enzymes were able to do the action of converting protein A to protein B. So using this information, they tried to understand what was the relationship between these genes and these enzymes. How might they be able to do that? And they were able to also take advantage of the fact that they understood that certain things made mutations in genes. So if you take an animal or sperm from an animal and you shoot x-rays at it, you make mutants. And so taking all of these pieces of information together, they designed an experiment to be able to demonstrate that one gene makes one protein. And that's what's shown here. So what you can see here is this is a tube, a little test tube. There's some food growth at the bottom, and then a little bit of yeast growing up on top, right? And if you take wild-type yeast, just normal, happy Neurospora, it's actually a mold, sorry, it's a mold, not a yeast. If you take this and you grow it, normally it doesn't really matter what food is in that test tube because the organism is able to make all the nutrients it needs. So if you, as long as you give it basic food, it's able to grow, and you can see that here. So when you have just the most minimal food, the little red spot is there, so the organism is growing. If you have minimal food and you add to that ornithine, then you still grow. If you add a little more, something else, this is called citrulline, then you grow. And in this one, it's basic food plus arginine. So a wild type, perfectly normal Neurospora doesn't care if you add vitamins or nutrients to the growing material. But now if you take one of these strains of Neurospora and you shoot x-rays at it and you make a mutant and you grow that mutant and you see, can this mutant grow? Now in certain types of food, what you'll notice is here, this is, again, that totally non-supplemented food. So the most minimal food, it doesn't grow. Okay? Question, yeah. Uh-oh. It is highly possible that did not, oh, you're right, look at that. Well, see, this is why we're doing a test run. If you guys are at home and it's not working, because I will do this during lecture time, there's a chat. I will activate the chat button, so you can just chat me until I catch on. Which is the other reason why I didn't tell you before you showed up for class, because I was like, watch this not work at all. Okay. Oh, somebody was raising their hand. Oh, wow. Holy, this will be easier when I'm not trying to talk to you and stare at the computer. All right. So, thank you for letting me know. Anything else I should, while you have my attention? Okay, good. All right. So in this circumstance, what we can see is that this mutant strain will not grow in minimal food. It's missing some key ingredient it needs in order to grow. If you add ornithine, it's still not going to grow. So ornithine is not the thing that's missing in the biological process. If you add citrulline, it does not grow, but if you add arginine, it's able to grow. So in this case, arginine is necessary in this mutant to grow, okay? Now there's a different mutant. This is a different strain. This is a strain now we are calling mutant strain 2, and mutant strain 2 won't grow in minimal food, it won't grow in ornithine, it will grow in citrulline, and it will grow in arginine. And here in mutant 3, it won't grow in minimal food, but it will grow in any of the other modified environments. So that was the experiment, and the purpose of this experiment was to see if we had one gene makes one protein. And this is the biological pathway that they were looking to try to solve. So we start off with the precursor. This precursor is converted by enzyme A into ornithine, and then converted by enzyme B into citrulline, and then with enzyme C into arginine. And the hypothesis was that there's gene A, it made enzyme A, gene B made enzyme B, gene C made enzyme C. And so if you look at this data, then the next question becomes, what can we learn with all of this information together? So let's take a step back. We know that under normal circumstances, the precursor can be converted all the way to arginine, which is what the cells need. And so mutant 1 cannot grow in anything except for arginine. So I want you to take a moment and think about which of these parts of the pathway may be broken, not working, interrupted, which gene might be mutated so that you have to add arginine in order for this Neurospora to grow. Yeah.
Gene A and B were mutated?
So if gene A is mutated, you cannot make ornithine. If gene B is mutated, you cannot make citrulline. If we were to provide citrulline in that circumstance, enzyme C is working. So could citrulline be converted to arginine? No. If enzyme C is working, if this last step is working, can we do this last step? Yes. Yes. So that means that in this case, We need a functioning enzyme C in order to do the last step of the conversion, right? So in order to produce the arginine, we need a functional enzyme C. So we can tell that mutant 1 has no functional enzyme C. Now, maybe it also has messed up everything else, but we know for sure that it's not going to be able to complete this last step, right? So what about this strain 2? This mutant strain 2 can grow only once you add citrulline, okay? Now, if we add arginine, it grows. That's fine. We just gave it the last step. If we add citrulline, it grows. But if we add ornithine, it does not grow. So again, take a moment. Which gene is likely mutated in strain 2 such that we see these results? So you think gene A is mutated. So if gene A is mutated and we provide citrulline, will we be able to convert, when we provide ornithine, right? That's this example here, right? Then we should be able to convert ornithine to citrulline to arginine. So in the case of strain 2, gene B is likely to be the mutant gene because if we add ornithine, we cannot make citrulline. But if we add citrulline, we can grow because we can make our own arginine. And if we add arginine, we can grow because we have all of the ingredients that we need. All right? So what I need you guys to do at home is do that same activity now for mutant strain 3. What is the probability, which of these genes is not functioning? Which enzyme is likely not present? And I can tell you that 90% of the time, this is a test question. Okay? So some variation of, here's the pathway, which enzyme is not working, how do you decide that is often a question that I ask. So the summary of the findings or the interpretation are that there is a single gene, which we are calling gene A, which makes enzyme A, and that converts the precursor, which is found in the minimal food, to ornithine. And then ornithine is converted now by the product of gene B, which is enzyme B, into citrulline, which is converted by the product of gene C, which is enzyme C, into arginine. And this generated this idea that we have one gene makes one enzyme. Now, one of the things you need to keep in mind is when this hypothesis was made, we didn't know that sometimes an enzyme was made out of multiple proteins. Okay? Now, you guys got to think all the way back to your second, I think it's your second lecture. Maybe it was your first lecture. What is it called? What is the level of structural organization when you have an enzyme or a protein, a complex protein, that has more than one polypeptide in it? Quaternary structure, right? So the quaternary structure is, by definition, when multiple proteins come together. And so what we would now say is the one gene, one polypeptide hypothesis. Because if one of those polypeptides is defective, is mutant, then the enzyme probably does not function correctly. So we've updated this now to include that additional understanding. Okay, so then how do we get from the DNA all the way to the protein? And we do that using what is referred to as the central dogma of molecular biology. And that is just this, that DNA is transcribed into RNA, and RNA is translated into polypeptide. Okay, so DNA to RNA to protein. And it's important for you to keep the two terms, transcription and translation, separated. These are key vocabulary words. You're going to see them often. You should be able to use them effectively in sentences to describe the images from the textbook. Transcription is when you make a copy of the DNA into a complementary sequence of the RNA. And translation is the conversion, or to actually, in fact, translate the language of RNA into the language of the amino acid. So we now know that in fact, there are some exceptions to the central dogma, right? So it used to be that we thought DNA to RNA to proteins. But we now know that because of studying viruses, that sometimes we see cases of other scenarios. And so that's what's shown here. So here we have RNA making RNA, making a polypeptide. And in fact, the coronavirus that we were just talking about before is an example of an RNA virus. It's a single-stranded RNA virus. It makes a double-stranded RNA, which then is used to make single-stranded RNA, which is then used to make this polypeptide. Now, there are also viruses, which are RNA viruses, which then convert to DNA. Though that DNA can also be integrated into the cells in your body and then converted back to RNA, and that RNA is then used to generate that polypeptide. Examples of this are HIV, and they are called retroviruses. And by understanding how this category of viruses work, we've learned a lot about how to make antiviral medications that specifically work against this type of virus. So again, in the context of the coronavirus, right now we don't have a treatment for the coronavirus. Specifically, we can treat your symptoms, but not the virus. We don't have a vaccine yet because the federal government cut funding to that vaccine program about 4 years ago, three and a half years ago. The retroviral drugs that you may have heard of that help prevent viral infections are targeted against retroviruses, not against RNA viruses. So right now, we treat you for pneumonia, we treat you for the cold symptoms, but we can't actually give you any medication to help with your viral disease, with one exception, but we'll talk about that in a few weeks. Yep.
So this only can occur in this process, these processes can only occur in viruses, or can they occur in
Right. So the way that these processes work in other cell types because of the virus. So a virus is not able to replicate its genetic material on its own. A virus is only able to do so in another cell type. So a virus, some people would argue it's not a living thing because it's not able to do the job on its own. It is a parasite. It infects the cells of another organism. That might be a bacteria or it might be us. And once that genetic material is inside us, it will replicate either using the cell's machinery or DNA that makes its own proteins that then it can use inside the cell. And in the second half of today, we're going to talk about how that happens, because conveniently we're talking about viruses in the second half of class. Yep?
So where did these viruses come from? Where they evolved from?
Very good question. Where do viruses come from? There are a lot of really interesting ideas about viruses. There are, in fact, what are referred to as giant viruses, and giant viruses look something like a mini bacteria. And so it may be that these are just variations of bacteria that got smaller and smaller and smaller until it became the most minimal unit. We don't really know. I know that they also spent some time talking in B106 about that. So, yeah.
Are viruses the only exception to the DNA, RNA, protein scheme?
That's a very good question. So are there other examples in which we see atypical behavior? And the simple answer in biology is there are always examples. For the purpose of this course, we're going to say no, because the examples are very, very rare and very poorly understood. Okay? So I would say really this is something we see very commonly in viruses and basically not in any other circumstance. Yep? Good question. So the question is, what do we know about the evolution of viruses? It's very hard to find evidence of viruses in the fossil record. They are microscopic. And so the hypothesis is that they are probably as old as bacteria because bacteria have very ancient viruses and they probably evolved in tandem. Yeah, there was a few more questions.
So those two processes are not occurring in the virus itself, it's occurring in the cell.
They are, that is correct. They occur in a cell that is infected with a virus. Okay. And there's a special enzyme that does this particular step of going from RNA to DNA, and it's called reverse transcriptase. And it does what is called reverse transcription. So we need, in order to go backwards in this, we have a special enzyme that carries out reverse transcription. Okay. So, I hate word slides. They're in here because it helps you guys because none of you write down everything I say. But here's what the idea behind this slide is. The next set of things that we're going to talk about is how a cell goes about the process of converting the DNA information to the RNA information to the protein information. And there are a series of hypotheses that were developed over the years to explain what happens. And so the first is called the sequence hypothesis, and it was made by Crick, of Watson and Crick, that developed the structure of DNA. And it basically was this idea that the sequence of the genetic material determined the sequence of the amino acids. He also developed a theory called the adapter hypothesis, which said, okay, so we have two languages, right? We have this nucleic acid language and this nucleotide language, and how do you translate them? And he imagined that you translate them through an adapter. It's kind of a code reader. And these are referred to now as transfer RNAs. So tRNAs are the translator in that they have the ability to identify the DNA sequence or the gene sequence, okay, and the protein amino acid that is associated with that. So they're kind of the code breaker. The tRNA molecules themselves contain what is called an anticodon, and the anticodon is complementary to the codon of the messenger RNA. And the tRNA is going to have to be very specific. So for every amino acid, there's one tRNA, and they're going to line up together so that it's able to do the translating. And we're going to look at how each of these play a role in the steps moving forward, but this is the vocabulary that you need. Okay, so where are these processes taking place? First thing to know is that in a bacteria, it's different than in a prokaryote, like a bacteria, it's different than in a eukaryote. Why? Exactly. So a eukaryotic cell has a nucleus. A prokaryotic cell does not. The genetic information, the DNA, is contained inside the nucleus and in a eukaryote. And so in a eukaryote, we're going to be converting the DNA sequence to the messenger RNA sequence inside the nucleus. In other words, we're going to be doing transcription inside the nucleus. And then that genetic material in the form of messenger RNA is going to leave the nucleus to enter the cytoplasm, and that is where translation will occur. In a prokaryote, it all occurs in the one place because there's no nuclear envelope. Okay, so what are the structures of the messenger RNA, of the DNA versus RNA, etc.? So RNA stands for ribonucleic acid, whereas DNA stands for deoxyribonucleic acid. And they are almost always single-stranded. So DNA is usually double-stranded. RNA is usually single-stranded. It has a ribose for its sugar backbone instead of a deoxyribose. And it contains uracil instead of thymine. So everywhere you would have a T in the DNA, an RNA would have a U as its equivalent, and you can see structurally they're very much the same. Here's thymine, and here's uracil. And we're going to see that sometimes a single-stranded RNA will fold up on itself and make really wacky shapes. There are three kinds of RNA involved in the synthesis of proteins. If we have time at the end, we're going to talk about some more recently discovered RNAs that do some really interesting things. But these are the three RNAs that are involved in the synthesis of proteins. They are the messenger RNA, also known as mRNA. They carry the genetic sequence. There's the transfer RNA, the tRNAs. Those are the adapters that translate the mRNA and the protein. And then ribosomes, which actually make the protein, contain a series of messenger RNAs, which are important for how a ribosome works. And these are called ribosomal RNAs. If you stay in the biology major, when you go on to other classes, you'll learn a lot more about ribosomal RNAs. They actually do some really cool things. But for today, they're a part of the ribosome. That's all you need to know. Okay, so what are the components of transcription? So if you want to take that DNA sequence and make messenger RNA, what do you need? Well, first you need the DNA. Okay, so the DNA template starts off as this double-stranded DNA sequence, and we're going to use one of those as the template from which the RNA will be made. Then we need the nucleotides that are actually going to make up the polymer that is messenger RNA. Those nucleotides are going to be ATP, GTP, CTP, and UTP, because remember, we're not having TTP, right? We're having UTP. And these are the substrates that are going to make that messenger RNA. And the last thing that we need is an enzyme to carry out this function. And this is called RNA polymerase. And that should sound similar to you to the DNA polymerase. But in this case, instead of replicating DNA, we're making RNA. Question?
What's the difference between a nucleotide and a nucleoside?
Good question. So a nucleotide versus a nucleoside, it depends on how many phosphates are bound to it or not. So in this case, when we're talking about ATP, it's a triphosphate. It has 3 phosphates associated with it.
And the nucleoside only has 1?
Correct. Yeah.
Oh.
Okay, so RNA polymerase, it has that term A's at the enzyme that tells us that it's an enzyme. Anything that says A's at the end, it's also composed of proteins. And I always like to show you a little bit of what they look like for real, because this blob over here is how we represent it in our figures. But this is what it actually looks like. And so you can see in blue, that's the DNA strand. In green is the RNA. And then in yellow is the RNA polymerase. And it looks very much like a hand with the thumb kind of poking out, right? So that's where the DNA and RNA interact with each other. And RNA polymerase is an enzyme that catalyzes the synthesis of RNA. It takes that double-stranded DNA, opens it up, uses it as a template to make messenger RNA. And interestingly, the RNA polymerase does not require a primer. So you may remember from DNA replication that in order to make DNA, you need a 3' end. That 3' end is then your starting point to make more. This can work without it, right? So you can just make your RNA without it. In addition, RNA synthesis does not have a proofreading mechanism. So I don't know for sure if Dr. Tajirian talked about the proofreading that occurs, yes, during DNA replication. Why is it important that there's proofreading during DNA replication? You got lots of hands. Good, I like hands. Yep. No, you get it. Yep.
So when it needs to carry out its function, it doesn't want to do the wrong thing, so to make sure that it's always right, because there's a lot of mistakes it can make. And
That's right. We want fidelity. If you make a mistake in replicating your DNA, that's inherited to the next generation. So you really want to make sure that your DNA is exactly like your parent DNA. So RNA polymerase does not have the ability to edit. Does that matter? Is that important?
Yeah.
It's a little bit important. Okay, why is it important? Do we want it to have the editing feature, or do we not really care?
one and two.
Well, I would say that you wanted to simply because RNA is what makes proteins, and proteins are what forms all the functions in the cell within the body.
Right.
Therefore, there's enough stuff like that.
So I heard a lot of people saying that it would be really important to do the editing feature. And it probably could be, absolutely, right? Except it doesn't have it. So the cell doesn't seem to think it's really important. So the reason it probably doesn't think it's very important is for several reasons, and we're going to talk about them as we go through. But one of them is that we make lots and lots of copies of the RNA. And so if one of them has a little error here or a little error there, it's probably okay. Another reason that it's probably okay is because, as we're going to see, the translation code is really flexible. And so it's possible for the small error in that messenger RNA to be invisible when we make a protein. It's also possible for the protein not to be changed because what you may know is that the shape of the protein is the thing that's really important for the protein to function properly. So I'm sure you talked about different types of inhibitors and different ways that the proteins change shape to be activated or inactivated. It turns out that if you trade out one amino acid, if it's a small charged amino acid for another small charged amino acid, the shape of the protein and then the function of that protein is probably okay. So there's a little bit of flexibility in that error message, in errors in that initial messenger RNA, and probably the energy that needs to get put into doing editing is too much energy to justify the work. Question?
So you said the RNA polymerase makes the RNA, but does it open up the DNA strand?
Good question. So what else is happening in this process? So there are a gazillion enzymes that we are not talking about. So we're going to talk about a few of them and go through each step carefully. But yes, you need something to open up that DNA. The RNA polymerase is not doing that on its own. Okay. So the process of making messenger RNA, again, is called transcription. It occurs in the nucleus, and it has 3 steps. It has initiation, elongation, termination. Okay? Now, it is very important that for the next few slides, you're paying careful attention to whether we're talking about initiation, elongation, or termination. And I can tell you, we get these mistakes on the tests all the time. I'm going to ask you a test question, and it's going to be exactly the same test question, except in one case, it will ask you about initiation. In another case, it will ask you about elongation. And the answer to those questions are different, right? Because the processes are different. So when you're studying this part, make sure you're able to distinguish those 3 steps and what's happening at each of the steps. And equally important, when you get to the test, read the test question carefully, because it's going to be about one of those 3, and you want to make sure you're paying attention to which one. I know it seems silly sometimes I say these things, but I have a number of students who've come back to me and are be like, but I answered that question right. And I'm like, no, I'm sorry. You answered it for initiation, but in this case, it was a different step. So just a heads up. Okay, so what's actually happening? The first thing that we need to figure out is where in the DNA are we going to start making the messenger RNA? Because not every part of the DNA has a gene in it, right? So every gene has a place where we initiate the messenger RNA synthesis, and this is instructed by the promoter. So there's a promoter at the beginning of every gene that says, I'm a gene, come over here and make me, right? And so I kind of like to think about it like that, like it's a party planner, and he's like, this is the hot spot. Come hang out with me. We're going to make some cool stuff. So the promoter is a special sequence of the DNA, and it helps bring the RNA polymerase and the other things necessary to make RNA into that section. Okay, so the RNA polymerase is going to bind that promoter, and then that promoter is going to tell the RNA polymerase which are the 2 DNA strands to start on, right? Because they have different base pair sequences. They're complementary, but one is the correct one and one is the not correct one, okay? So the promoter is going to help tell the RNA polymerase which is the correct one, and it's also going to identify the initiation site. So we can see here is RNA polymerase. It's checking to see whether or not we're in the right place and which is the right strand. So now we've opened up that DNA. We see that symbol of that hand shape for the RNA polymerase. And here in purple is the representation of the initiation start site. So this is where we're going to start making that messenger RNA. Here's the rest of the DNA template. And then over here we have a termination sequence, right? So first the RNA polymerase finds the initiation site. And now during elongation, the the messenger RNA is going to be synthesized. And so what you're going to see here, this is the strand of the DNA that's being replicated, and this strand is 5' on this side, 3' on this side, 5' on this side, okay? And that's because DNA is reverse complementary, and as you can see, the RNA starts off with a 5' end. So in order for this to be a 5' end, the sequence that's using as a template must have the 3' complementary sequence on this side. So here we told you we needed our template, we needed the enzyme, and we needed the nucleotides. Here are the nucleotides. And you can see these nucleotides are going to enter the growing replication site, our transcription site, and now we're going to make that messenger RNA. And we're going to move along the DNA template until we reach an endpoint. So a couple of those notes are written out for you. RNA polymerase is going to unwind the DNA about 10 base pairs at a time, and it's going to do that in the 3' to 5' direction based on the template that it's using. And then the RNA polymerase is going to generate that anti-parallel complementary sequence. And then the RNA polymerase is going to not bother proofreading, because it's better to just crank out lots of RNA and trust that it'll all be okay. Now, third step, terminate. There is a special sequence in the DNA that says you have reached the end of the messenger RNA. It turns out there are several different ways that cells do this. And again, if you stay in biology, you can take an entire semester class just on this. Even when I was an undergrad many, many years ago, I took a class that was literally an entire semester on transcription and translation. So this is just a taste of, if you think this is really awesome, you can go learn more. Question? Good question. So if you choose either one, do you get the same protein sequence? And the answer is no. You don't get the same messenger RNA sequence, so you don't get the same protein sequence. So one of them is going to be correct and one of them is not. Yep. Don't you have a few codons? I'm not sure what, I think your language isn't quite accurate. So a codon is a 3 nucleotide sequence that says that does the translation for you. The easiest thing for me to do is during the break, I'll put a sequence on the board, the DNA and then the messenger RNA and then the codons, and I can show you how different they would be. But that's also an exercise you guys can do on your own. So if you were to make up a DNA sequence, double-stranded, and then you can actually calculate out what would be the protein you would produce from either strand. But the answer is they are very different, and so one of them is going to be useful and one of them not as much. Question?
So which strand is the RNA?
Right. Good question. So the RNA that is made is 5' to 3'. So the template that it uses on the DNA strand has to be what? If it's 3' to 5'. Correct. Yeah.
Are there any cases that it does it the other way around from 3 to 5?
So, couple of clarifications there. Mechanistically, no. You will never have the ability to make the DNA or the RNA in the opposite direction. That being said, the way that this is visually represented, it implies that it always goes on the same one. So if you are using, not sure which one is, we're going to call this strand over here the 3' making its way to 5' over here. So that's going to make RNA in this direction, okay? You can just as easily have the gene be on the opposite strand, but if it's on the opposite strand, it's going to need to go the other direction. So they always show things in the way that we would read them in English as going from left to right, but the cell may have some genes go this way and some genes go this way, as long as it's using the 3' to 5' sequence as a template. The other thing to keep in mind is that the sequence of the messenger RNA that's 5' to 3' here should be the same as the sequence of the DNA on the complementary strand, right? So we're replicating the 3' to 5' strand, but the sequence of this messenger RNA will be the same as the sequence of the DNA's 5' to 3' strand.
So how long is the sequence of one gene?
Very good question. How long is the sequence of one gene? It totally depends. You can have a sequence of a gene that's only 100 nucleotides long. You can have the sequence of a gene that's tens of thousands long. And that depends in part on whether there are introns, which are going to get removed, and we're going to talk about that. And it also depends on how long the protein is. Some proteins like insulin are very short. Some proteins like myosin are very, very long. So you just have, depends on what you're making. Yep.
So when you're doing the RNA polymerase and replicating, are you always making messenger RNA? And like, is the transfer RNA and the
Good question. So we talked about transfer RNAs, ribosomal RNAs, and messenger RNA. So the cells are also able to make, in a very similar mechanism, these other types of RNAs. They have specialized RNA polymerases for that process. So the generic process is the same, but some of the regulation and some of the details are a little bit different. Okay. So we reach a termination sequence. That termination sequence lets everything fall apart. The DNA reforms the double helix. The messenger RNA is now separated and available. And there are additional proteins available to help this process terminate. All right, so now we need to do translation, right? So we need to convert the language of the RNA to the language of proteins. And the genetic code is composed of these short 3 nucleotide sequences called codons. And each 3 nucleotide sequence translates into one of the amino acids. There are 20 amino acids that are coded for in the DNA. You may see that there are, in fact, 22 different kinds of amino acids found in biological systems. Those other 2 are not encoded by the DNA. They are modified later. Okay, so 20 is the number that is actually encoded in the DNA. And so the DNA has 4 letters, the RNA has 4 letters, but amino acids have 20 words, and it's a codon sequence of 3 bases that identifies the specific amino acid that you need. We know that this is the case because of a series of experiments that were done where they literally, in a tube, tried to make different strings of proteins. And what they found was that if you put in a whole string of U's, that you would get a protein that was phenylalanine, phenylalanine, phenylalanine. If you put in a string of C's, then you would get proline, proline, proline. And so if you changed the sequences of the RNA in certain ways, you could predict which amino acid strings you would get out. So another question that comes up often is how many different codons are there? So we know that there are 20 amino acids. We know that there is a letter code of 3 nucleotides to give us the codons. But it turns out that this statistically gives us 64 possible codons. And we know this because there are 3 positions, right? The codon has 3 positions, and each position has 4 possible options. So if we multiply position 1 with 4 options by position 2 with 4 options, multiply again by position 3 with 4 options, you multiply 4 times 4 times 4, you get 64 codons. This is going to feel like a silly little fact, and the reason I emphasize this is that this gives a whole lot of flexibility to the messenger RNA to protein translation process. If this was different, if it was only a codon of 2 nucleotides, or if there were 5 options for the number of nucleotides, that would give us a really different fundamental structure to DNA. So it's important to understand why this is the case. Question?
So do we use each codon?
Good question. So yes, each codon is taken advantage of, and we're going to talk about them in next. Next, look at that. Okay, question.
I wanted to ask something about that. So the experiment says that they added an artificial RNA, but then the slide says that they used artificial nucleotides instead of the RNA.
So it's not artificial in the sense that it's fake. It's artificial in that it's not found in a cell. It's one that's made in a lab. Okay, so at this point, because technology advanced, you can literally just call up a company and say, I want RNA with this sequence, and they just send it to you. Okay, so these are all the possible codons and the nucleotides that they or the nucleic acids, the amino acids, that they work with. So here's how to read this chart. There are 3 letters in the codon. The first letter is indicated here, U, C, A, G. The second letter is indicated here, and then the third is within the box. So you'll see first one is U, second one is U, and then the third one is going to vary. And what you're going to see is that this is a sequence that is both specific but redundant. So it's specific because every single amino acid is encoded for by a unique codon, but that it can be there can be many different codons for that amino acid. So here, for example, is leucine. You can see right away there are 6 different codons that all will give you leucine. And this is, again, one of these reasons why making RNA perfect doesn't matter, because hopefully the mistake that was made was in that third position in the codon. And you can see that as long as the first 2 letters in the codon are C and U, you're going to make a leucine. So it doesn't matter if that third position is wrong, you're still going to make the same thing. So there are a few special codons that do not behave the same as others. One is methionine. The codon for that is AUG. All proteins start when they are originally made with a methionine. It is how the cell knows that that's the beginning of where you make the protein. So there's a small portion of the messenger RNA at the beginning that doesn't make protein. It's just holding everything in place. It's providing some information. And then you have an AUG. And that first AUG is called the start codon, and it makes methionine. Question?
So like codon is like translation or
You okay? Yeah.
This all is read in the ribosomes.
So this is all done within the ribosomes. We're going to show you how the ribosomes work in just a moment. Okay, so then there are also 3 codons that do not encode for an amino acid, and these are the termination codons. They are listed here. When you get to a space in your messenger RNA that has one of these sequences, UAA, UAG, or UGA, there's no amino acid that is encoding for that. You get termination, the system falls apart. You do not need to memorize this at all. If I'm asking you to do a synthesis, if I say, here's a messenger RNA or here's a piece of DNA, make me a protein, I will give you this chart, okay? So that does mean you need to know how to read this chart, take advantage of what's in the chart, but no, you do not have to memorize the chart. The only thing I would encourage you to know is that the start methionine sequence is AUG, right? So that one start site, you need to know that it encodes for a methionine. Okay, so the messenger RNA that is produced needs to be processed often before it is mature and ready to make the protein. So this is because if you look at the sequence, here's a gene, here's that promoter, here's the start site, which is where the messenger RNA will first be made. And then inside that, there are what are called introns and exons. And you have to splice the pieces together so that only the pieces that are necessary to make the protein get joined together. And so you see the dark green boxes, these are exons. They will be spliced together, removing the introns to make a correct and final messenger RNA. So introns are cut out, exons are removed from the pre-messenger RNA to make the mature messenger RNA. If you mess this up, then you're going to have a very different protein. Question?
Where do the introns go?
Where are the introns? They just get degraded. They're just reused. Yep.
So what shows like, oh, here's an intron?
How does the cell know that there's an intron in that position? There is a special DNA sequence and therefore a special messenger RNA sequence at the junctions between an intron and an exon, and we're going to look, if we have time, at how that works. But there are these things called SNRPs, which recognize those sites, cleave them at that site, and then help the two exons stick together. Okay, so just a reminder, different things are going to happen in different places if you're a prokaryote versus a eukaryote. Prokaryotes, it's all taking place in the cytoplasm. Eukaryotes, we're going to make the messenger RNA in the nucleus, and we're going to cut out those introns in the nucleus, and then we're going to process that messenger RNA. We need to stabilize that messenger RNA. We want it to be happy in the cell for a good long time. We do that at both ends. So the 5' end gets what's called capped, and the 3' end has a series of nucleotides, just the nucleotide A, added at the other end. And that helps keep that messenger RNA in the cell for an extended period of time. I did have a question. Yeah? All right, if you think of it again, we'll come back to it. Okay, so here's what that it looks like. So the messenger RNA will have a 5' cap, which is a modified form of the GTP, and will have a poly A tail. So if a protein needs to be present for a long period of time, if we want that messenger RNA to be present in the cell for a long period of time, we can do that by stabilizing that messenger RNA with these two additional modifications. Okay, so this gets to the question of how do we cut out the introns? This process is really fairly simple. So here we have the RNA, the messenger RNA, immature messenger RNA has exon, and then intron, and then exon, and here we have these SNRPs, these RNPs, and they recognize the site where the RNA needs to be cut, and they will generate a small little loop out of that intron, and then they will excise the intron, reseal the messenger RNA so that it is a mature messenger RNA. And this has to occur correctly, otherwise you have a messenger RNA that has the wrong sequence in it. So now this mature RNA is able to leave the nucleus. It's going to leave the nucleus through a nuclear pore, okay? And there's a cap binding protein that's able to bind that 5' end of the messenger RNA, and it's through that we're able to properly remove the messenger RNA. So we don't want to remove messenger RNA from the nucleus that has not been matured yet, but you don't get that 5' cap until it's ready. Once that 5' cap is on, that's the signal to move through the nuclear pore and out into the cytoplasm. Okay. I'm not going to read this slide, but it basically just says, yet again, in 4 different ways, that the tRNA is the translator molecule, and so you need the correct tRNA sequence with the right anticodon tagged to the right amino acid so that the translation occurs correctly. Question?
So why do we add the poly A tail?
So the poly A tail helps prevent the RNA from being degraded. There are lots of enzymes that will chew the end of the messenger RNA, and if they just chew A's off the end, it doesn't matter, no one cares, because it's not getting into the protein coding sequence, but eventually you start to chew off nucleotides that are important for the messenger RNA. So the longer the poly A tail, the more stable that messenger RNA is inside your cell.
And once the 5' cap is on the RNA, it's ready to leave?
That's correct. That's the indicator that it's mature. So this is what a tRNA looks like, and in classic textbook fashion, they show it to us several ways. Question?
Yeah, this is from a little
Sure.
What is the purpose of introns?
What are the purpose of introns? Very good question. So introns allow several things. Within introns, there are regulatory elements that tell genes when they should be made into messenger RNA or proteins and when they should not. And we're going to talk about that a little bit later. They're also helpful in what is called alternate splicing, which we're going to talk about also in a little bit. Alternate splicing allows a single messenger RNA sequence to be pasted together in several different ways to make very related proteins. So there'll be circumstances where you want exon 1, 2, 7, 9, 11, and then another circumstance where you might want 1, 2, 3, 4, 5, 6, 7, 12. Okay? And so you can paste the different exons together from that individual messenger RNA, and that allows you to make variations of the same protein. Okay, so this is a tRNA. You can see that it has this RNA sequence. It's single-stranded in some parts and double-stranded in others, and has these little loops. This loop at the bottom here is where your anticodon is. That's that most important sequence and during the translation. And then the other thing that you have is at this 3' end, this is where the amino acid attaches to the RNA. And so what's really important is that we have what is called high fidelity in the right amino acid binding to the right codon, anticodon, to the right tRNA. Otherwise, our translation gets messed up. Okay? So there is a special set of enzymes that make sure that every tRNA is correctly tagged with its correct amino acid. Again, high fidelity, and that helps ensure that the proteins we make are the correct proteins. This is what it looks like in 3-dimensional space, and this is what it looks like in a more ball and stick model, and this is the icon that is used in the textbook to show you that tRNA. Question?
So when the protein is being made, you want the ribosomes to read the RNA 5 to 3'. That's why it's going to 3' first.
Yep. Okay. So in our DNA, we have our gene. Okay. We read in the DNA template, the codon is going to be GCA, right? And that's going to make an arginine in our protein. The complementary strand to that template strand is going to be the messenger RNA. It has a 5' end that's going to say then CGG. That's the reverse complementary to the template. And then there's the anticodon sequence in the tRNA, which is now going to go 3' to GCC and then end in the 5' side. So again, test question. Perfectly reasonable for me to give you a DNA sequence, ask you what the complementary messenger RNA sequence is, and then to ask you what the anticodon sequence is. So those are good things to practice and make sure that you really understand. They're not good things to memorize, because that's a lot of memorizing rather than thinking. Okay, so we really want to make sure that the correct tRNA, by which I mean that it has the anticodon, is flagged with the correct amino acid. And so there are 20 separate enzymes to do this. Every single amino acid has a unique enzyme. Now, not every single anticodon, right? So we have 64 codon possibilities, some of which, right, 61, make an amino acid addition into the messenger RNA, but we have 20 amino acids and 20 enzymes. So the enzyme can recognize all of the tRNAs that encode for the same amino acid. And so here we have our enzyme. Okay, this is, they're called aminoacetyl tRNA synthetases. And you're going to see a particular amino acid is going to enter the spot. Okay, so in this case, the example they're giving is alanine. In this process, the energy from ATP is utilized, and so now we have an AMP sitting in this position. And now the tRNA is going to come in. As that tRNA comes in, now we change the configuration of this enzyme that causes the release of this AMP. Whenever you release something, it changes the shape, usually allows for an energy transfer and a bond to be made. And now we have the correct tRNA labeled or charged is what it's called with the correct amino acid. So in this case, alanine is going to be added to that tRNA, and now you have a charged tRNA, in this case charged with alanine. Yeah.
So those enzymes are specific for amino acids?
They are amino acid specific. There are 20 amino acids and 20 enzymes. All right. Ribosomes. So ribosomes are going to be the machinery that does the translation. Ribosomes contain two major sections, the small ribosomal subunit and the large ribosomal subunit. Each of the subunits contain a bunch of RNAs and a bunch of proteins. There's a ton of stuff in there. There's a ton known about how they work. We are doing the B105 version of this. If you're really excited to learn more, let me know. I'll happily teach you more, but just go in textbook simple today. So the things that you need, they're going to be in this ribosome 3 parts. There's going to be the A site, the P site, and the E site. E stands for exit. There's going to be a place for that messenger RNA to bind, and there's going to be a place for that charged tRNA to come in and identify the codon and make sure that the right protein is made. The ribosomes are not going to be specifically designed for any one protein. So the ribosomes make all proteins. And the two halves of the ribosome, the large and the small subunit, are held together by ionic and hydrophobic forces. There are no covalent bonds between the large and the small subunits. And they exist separately, floating around in the cell when there's no messenger RNA in there ready to be translated. So they exist as two separate pieces. So again, we want to keep in mind in this context that there is, just like we saw with the synthesis of the messenger RNA, 3 steps: initiation, elongation, termination. Keep track of where you're at. And remember, in eukaryotes, this is taking place in the cytoplasm, just like in prokaryotes. And sometimes this takes place on the surface of the rough endoplasmic reticulum. Because if you remember way back to like lecture 2, when you looked at the parts of the cell, the ribosomes were the thing that made the rough endoplasmic reticulum rough. So the first thing that happens is the small subunit of the ribosome identifies a messenger RNA that's going to be translated. The start codon aligns and the RNA kind of sticks in the right spot. And so it'll make this small ribosomal messenger RNA component. The next thing that's going to happen is that methionine tagged charged tRNA is going to come into this complex. So remember that the start site is always AUG, always makes methionine. And once that happens, then the large ribosomal subunit can come in and complete the formation of the ribosome, and we can start synthesis. Question?
So that second step, that's the charged.
This is the this is a charged tRNA because it has the appropriate amino acid on it. In this case, the appropriate amino acid is methionine. It's an AUG start site. Now we've completed our step 1. Yep. Yeah, the Shinel-Delgarno sequence is the thing that allows the alignment to occur the way that it has occurred in this first section. Yes. Now we're going to go to step 2. So in step 2, we want to do elongation. What happens in elongation is the next tRNA has to come in. So here we see that tRNA spot in site A. So the first one is in the P site. The second one's going to come in to the A site. And it has to match, has to be the right one. So the anticodon from the tRNA identifies the codon and the messenger RNA. And now we get that charged tRNA in the right position. Once it's in that position, the ribosome is going to move one step down the line on that messenger RNA, and as it does so, it's going to form a covalent bond between the charged tRNA and the existing strand. So we have 2 charged tRNAs, they're near each other. We now transfer the first amino acid covalently to the second amino acid, and we shift down the messenger RNA. And so you can see here now there are 3 amino acids stuck onto this one transfer RNA that's sitting in the P site. As we ratchet down the messenger RNA, the tRNA that was in the P site is now in the E site. That's the exit site. It gets kicked off. It no longer has an amino acid. It floats off and gets recharged, and the A site becomes available for another amino acid charged tRNA to come in. Question? We're running out of time, so I don't want to stop the questions, but
What's bonding covalently?
so the only covalent bond that takes place is between the amino acids, and no additional energy is necessary for that because the energy necessary for that comes from releasing the tRNA and transferring that energy to the new covalent bond. Okay, so we now have a growing polypeptide chain. It could be 10 amino acids, it could be 1,000 amino acids, it very much depends. When we get to that endpoint, the messenger RNA is going to have a codon that encodes for a termination. Is there a tRNA for termination? No. So we have a termination site. A termination factor comes in and says, you're done, and everything kind of falls apart. And now we have the messenger RNA available to make another protein. We have the protein available to do whatever its function is. Now, in prokaryotes, there are a couple things that can happen. Multiple ribosomes can bind to the same messenger RNA, and so we can have what's called a polysome or a polyribosome, and that's shown in a real picture here. But the idea is that as the messenger RNA is being translated, the ribosome moves down the messenger RNA, and another ribosome can come in and start translating as well. So you can see multiple, it's just a way to increase efficiency, to make lots of proteins from the same messenger RNA at the same time. We can also have sequences in the protein that is being made that halt the process. So if a protein is being synthesized and it is supposed to be in the endoplasmic reticulum, there is a sequence that says, wait, stop, take me to my home. And then that sequence will carry the messenger RNA and the ribosome and the little tiny bit of protein to the docking site on the endoplasmic reticulum and allow then that protein to be synthesized and sort of spit into the endoplasmic reticulum. Usually when that happens, the little sequence that brought it there gets cut off because it did its job. And now you have a protein that is going to be inside the endoplasmic reticulum. And the reason that we have proteins inside the endoplasmic reticulum is that sometimes proteins need to be changed up a little bit. They can be phosphorylated. Remember, changing the phosphorylation state may make it active or inactive. They may be glycosylated, which is a fancy word for saying there were sugars added onto it. Sugars are really important on the surface of cells for identifying different cell types. They do some other things as well. And they may undergo what is called proteolysis, which is a fancy word for chopping up the protein into smaller bits. And those smaller bits may be necessary for the protein to function correctly. So that happens only inside the endoplasmic reticulum. Okay, so if we have normal DNA, no mutant, everything's happy, here's a DNA template. And now, again, this is the DNA template strand. The complementary strand would be up here, and it would have the reverse complementary sequence. That reverse complementary sequence is the gene. Okay, so we would have the gene up here. This is the template that is transcribed into the messenger RNA, which is then translated into the protein. We can see that it starts with the methionine. And now if we have a mutation, let's say here, we have a mutation where the A is converted, we have an A instead of a C. In this case, the A and the C results in no change in the amino acid sequence because we've changed the third position in our codon. That third position, as we saw with lysine, you can have any one of the four nucleotides, it doesn't matter. It's the same tRNA, the same amino acid. This is referred to as a silent mutation, no effect. Alternatively, we could have what's referred to as a missense mutation. So here, we convert, we have an A instead of a T at this position. When we do that, we change this amino acid. Now it's a valine, and that's going to change the function of that protein, potentially. And lastly, we might have a mutation that generates a stop codon. So here we have a mutation that generates a stop codon. We have a T instead of a C, and so now we have a truncated protein. So thought problem here, if the mutation, instead of being at the beginning of the DNA sequence, or therefore the beginning of the messenger RNA, was almost at the very end of the protein, almost at the very end of the messenger RNA, right? Would that be as bad as at the very beginning? Probably not, right? Probably if you make 90% of the protein, you'll be okay. Sometimes that's not true, but often that's the case. This is the worst, or one of the bad kinds of mutations. This is an insertion. So this is where you added a T into the middle of your sequence. Now by adding a nucleotide into the middle of your sequence, you mess up that 3-character codon. right? So just by adding this one letter into the DNA, now every single one of the codons after that is shifted by one. So it's called a frame shift mutation. You shift by one, and now every single one of these codons is wrong. So every single amino acid after that point is wrong. Now, these can have different consequences on the protein that's made. So if you have a silent, here's your normal mutation, or your normal protein. It has a functional protein. Here we have a silent mutation, as we've said. Silent mutation has no consequence on the protein. Here we have what's referred to as a loss of function mutation. So this is a mutation where by changing that one amino acid, the protein doesn't work anymore, okay? And then we can have what is referred to as a gain of function mutation. Based on the name, gain of function, what do you think a gain of function mutation might do to the function of a protein? Changes it. It gains a new function. And I think this is the last idea. This is just an idea that says that sometimes, in addition to a protein having a sequence that says go to the endoplasmic reticulum, you can have a sequence that says, oh, go to the mitochondria, or oh, go to the chloroplast, or oh, go to the nucleus. And the way that we test that idea experimentally is we literally just add that little sequence that says, hey, go to the nucleus, to the protein. We inject it into a cell, that's what's shown here, and we see what happens. So in this first example, there's a protein with the nuclear localization sequence. You inject it, and you can see all of the red protein is in the nucleus. Now, if you have that exact same protein, but you remove the nuclear localization sequence, which in this case is indicated by an NLS, nuclear localization sequence, now it doesn't go to the nucleus. In this case, we have a different strategy here. We start off with a protein that's normally found in the cytoplasm, and that's where we see it. Now, if we add a nuclear localization sequence, where do we expect it to go? Into the nucleus, and that's exactly what we see. Okay, so thank you for your patience. We will start again at 8:15, and I will probably cut the last little bit off the end of the second lecture. So 8:15, we'll start again, and I will pace myself.
Professor, I didn't get this last part.
All right, hang on.